this hydroxyl group is right here. That's the only one that's getting metaphor. Hydroxyl group that's counterized by the beta carbon does not get metaphor. So this enzyme, at least in the case of the benzene ring, is absolutely specific for that hydroxyl group. It's not absolutely for the substrate, but at least in terms of catecholamines and degradation products, it's specific for that hydroxyl group. Okay? So what's effectively going to happen is there's going to be a base in the active site of the enzyme. Okay? There's going to be a base, and it's going to deprotonate that hydroxyl group. Okay? And what's going to happen is those electrons between the oxygen and the hydrogen are going to attack this methyl group that's on S-adenosine methionine. And certainly, if you need help with the mechanism of methyltransferase, certainly go back and watch the video on that. But effectively, chemical and methyltransferase is going to catalyze the attack on that methyl group. And of course, you're going to have loss of the leaving group. And in the case of this enzyme, and all methyltransferases of this kind, the leaving group is just S-adenosine homocysteine. So we can say it's ado-homocysteine. And so you can say that this one is ado-met, right? Adenosine methionine or S-adenosine methionine. And so what you should get at the end of this, you should get a methylated oxygen that is meta to the beta carbon, right? I keep saying that because it's extremely important when you're trying to predict products on an exam, okay? And also keep in mind that at the end, we also should have our base in the active site that's protonated, right? Our base in the active site is protonated. And what will effectively happen is there's a theoretical step in which there will be a proton exchange with solution to regenerate the methylation of the enzyme. So I hope that gives you a little bit of intuition on what catechol and methyltransferase is actually doing. So what I want to do now is give a little bit of intuition on monoamine oxidation. For those of you who haven't seen this before, sometimes you'll see this as MAO, and oftentimes if you have an inhibitor of monoamine oxidation, they'll say MAO inhibitor, so that's kind of important to know. So MAO is monoamine oxidation. By the way, there are multiple monoamine oxidations that have various specificities for different neurotransmitters. We're just going to talk about it in general. Okay, now keep in mind, this R group right here, if this is a hydroxyl group, then that's characteristic of both norepinephrine and epinephrine in their metabolites, right? But if that R is a hydrogen, then that's going to be dopamine. And if that R was a hydrogen, then this R prime would also have to be a hydrogen, right? Okay. And what this enzyme is going to do is to use FAD in order to basically take this part right here, and it's going to oxidize it into a shift base, okay? Or to be specific, a protonated shift base, okay? And that's what the mechanism involves. Maybe one day we'll have an entire video on the mechanism. But suffice it to say for now, the mechanism is extremely complex and convoluted, and as we know, shift-base mechanisms are not exactly the easiest thing in the world. They get really long and hard really fast, okay? So we're not going to deal with that here, but suffice to say that group will become a protonated shift base. And then what's going to happen is we're going to have water that's in the active site, and the water is going to do a shift base hydrolysis, okay? And that oxygen, let me do this in green. That oxygen that's part of the water, that's this oxygen that's part of the aldehyde, okay? So that's what we mean when we get a shift base hydrolysis. So when we do the shift base hydrolysis, okay, we're going to get an aldehyde as a result, and this would be the aldehyde that's this right here, right? That would be these aldehydes right here, this one right here, and this one right here. So in the case of norepinephrine, that would be normetanephrine aldehyde, and norepinephrine aldehyde, that would depend on whether or not it's reacted with catechol and methyltransferase, okay? But in this case, we're assuming that it hasn't reacted with catechol and methyltransferase. But we did a shift base hydrolysis, and we're going to get an aldehyde as a result. I hope that makes sense, okay? And that's all part of the mechanism of the FAD-dependent oxidation. So if FAD is oxidizing this right here into a shift base, right, then FAD in the process had to get reduced. Okay, so in the process of doing this, we're going to get FADH2, and I hope that makes sense. Oxidations can't occur without reductions, right? So if we're oxidizing the catecholamine into a shift base, that's an oxidation, so FAD had to get reduced to FADH2. And again, the mechanism is very complex, very convoluted, and we'll look at that in another video sometime, okay? But then the shift base gets hydrolyzed into an aldehyde, and then you get this molecule right here, okay? Now, this molecule changes, okay? Now, remember what I said earlier, back to this picture, okay? Dopamine products change, right? Because remember, in the case of dopamine, this was a hydrogen, right? And you can make this case in all the other molecules. That's a hydrogen in dopamine. So that product is going to be different than norepinephrine and epinephrine degradation products, right? But remember how I said that in general, these products right here, these products are the same in norepinephrine degradation and epinephrine degradation. Well, let's think about why that might be. Well, they both have this hydroxyl group on the beta position, right? But remember that the part of the molecule that gets hydrolyzed off is this, right, the amine. Now, in terms of the actual degradation products, which is this, right, norepinephrine degradation and epinephrine degradation is exactly the same, right? And I think you can see that, right? But the amine that gets hydrolyzed off from the shift-base hydrolysis is different, okay? Now, we'll come back to that in a few minutes, but let's actually talk about what happens to FADH2, okay? The reason this enzyme is called an oxidase has nothing to do with the first half of the mechanism. In fact, the first half of the mechanism is just an FAD-dependent oxidation. And in the process, remember, FAD gets reduced to FADH2. Well, the oxidase activity is going to use molecular oxygen in order to oxidize FADH2 back to FAD. And in the process of this oxidation, oxygen gets reduced to hydrogen peroxide. Okay, so this oxygen is going to get reduced ultimately to hydrogen peroxide. Okay, and that, of course, regenerates the resting state of the coenzyme FAD. And then we're ready for another cycle. Now, the hydrogen peroxide is going to get released by the enzyme. And as we know, catalase is going to degrade hydrogen peroxide into oxygen and water. Okay, and I hope that makes sense. Okay, we've dealt with that in other videos. Okay, well, let's come back to the problem. Okay? In the case of dopamine, let's think about what this amine might be. Well, in the case of dopamine, what was our R prime? Well, in the case of dopamine, it was just another proton, right? So if we're dealing with dopamine, we'll abbreviate that GA because that's normally the abbreviation. But think about our amine product. Well, in the case of dopamine, our amine product is just going to be ammonia, right? Because that other R prime group was a proton, right? So in the case of dopamine, that's going to be ammonia as our product. And we know from our studies of amino acid metabolism that the ammonia will either get ligated to alpha-ketoglutarate to make glutamate, or from glutamate, ligated to glutamate to make glutamine, or if we're in times of amino acid metabolism, we're going to essentially put it into the urea cycle. So it really depends on the energy charge of the cell as to which direction we're moving. Okay, so the point is we're producing ammonia. Okay, now let's think about norepinephrine NE. That's normally how we abbreviate it. What's going to be our amine product? Well, when we hydrolyze off that amine in the Schiff-based hydrolysis, our R prime is also a proton. So if we do this process with norepinephrine, we're going to get ammonia as well as our amine product. But now let's think about epinephrine. What's going to be our product there? Well, what's our R prime group in epinephrine? Well, our R prime group is a methyl group, right? Remember that norepinephrine reacts with phenylethylamine and methyltransferase, which is a SAM-dependent enzyme. And so that R prime is a methyl group, right? So that means our product is going to be this. Let me do it like this. Let's do this. So H3N, and then a methyl group, right? So it has a positive charge at physiological pH. So we get methylamine. Now, we're not going to go into excruciating detail here about methylamine, but suffice it to say what's going
Buonasera, io sono Marco Cinca, produttore del Piccolo, vi porto i saluti del direttore Paolo Posolai, che questa sera purtroppo non ha potuto essere presente. Benvenuti a questo questa anteprima in realtà del secondo ciclo di Science on the City, che l'anno scorso mi risulta, io purtroppo non ho potuto assistere a Montevideo, ma mi risulta aver avuto un notevole successo. Non mi dilungherò naturalmente in ringraziamento cose del genere, solo credo si debba a nome, ma credo lo farà anche il professor Giacca, un grazie alla Regione che ci ospita e che contribuisce a sì che questa iniziativa possa continuare. Credo arriverà tra qualche minuto Marco Longo, che è il direttore centrale della sanità, e quando arriverà, se lo riterrà, potrà dire qualche parola. Io credo che possiamo entrare subito nel vivo. Gli ospiti li conoscete tutti: il professor Mauro Giacca, direttore dell'ICGED, direttore mondiale da 6 mesi, se non ricordo male, 6 o 7 mesi, il dottor Tutto, direttore sanità igiene della provincia di Trieste, della sanitaria della provincia di Trieste. E in collegamento via Skype la dottoressa Giorgia Sentini, triestina, che è un medico di Medici Senza Frontiere, che credo abbia avuto una recente, non so se ancora, una recente esperienza in Sierra Leone, e quindi, come dire, potrà raccontarci dal vivo, da fronte. L'audio non si sente, devo parlare più forte? No, la stavo presentando, stavo dicendo che ho avuto un'esperienza molto recente, credo terminata da poco, non so se ancora in corso in Sierra Leone, e quindi potrà portarci una testimonianza molto importante sul tema di questa sera. Io non mi dilungherò molto perché credo che qualche domanda poi la faremo, la farò io, la farò il pubblico, ma credo che sia opportuno che sia il padrone di casa, cioè il professor Mauro Giacca, a introdurre l'argomento di questa sera. Grazie molte, benvenuti a questa seconda serie di Science and the City. Vi ricordo brevemente di cosa si tratta: è una serie di incontri aperti al pubblico con l'idea proprio di raccontare storie di scienza al pubblico, e storie che toccano la vita quotidiana, e speriamo non la nostra in questo caso, ma che comunque sono molto presenti sui giornali, nelle televisioni, ma spesso non spiegate in maniera opportuna, quindi questa è un'occasione veramente per sentirvi raccontare da vicino cosa succede, anche per dare la possibilità chiunque di fare domande e approfondimenti. Questa è una serie di incontri che è stata finanziata dalla Regione Friuli Venezia Giulia, che ringraziamo moltissimo per la sensibilità proprio alla divulgazione in senso scientifico. Cultura, il nostro paese spesso vuol dire cultura, non si parla di cultura umanistica, ma è una secca un po' italiana, insomma, e penso che dovremmo tutti saperne di più di scienza, e quindi grazie per questa sensibilità. L'organizzazione di tutto il ciclo di eventi che avrà luogo in primavera sarà insieme con il Comune di Trieste, e il supporto economico del Rotary Club Trieste Nord, e la Media Partnership del Piccolo, che è anche un nostro partner molto importante. Sapete che l'ICGED è un organismo internazionale con sedi in 3 paesi, e esiste questa richiesta, è sempre più pressante anche a livello internazionale, di fare divulgazione scientifica, e quindi noi pensiamo di interpretare bene questo tipo di eventi, questo tipo di richiesta con questi eventi di Science and the City, questo è il cartellone dell'anno scorso. Vi faccio vedere questo perché tutti gli eventi, compreso questo, sono registrati e sono poi prodotti in un formato che è scaricabile gratuitamente dalla piattaforma di iTunes. Se voi avete un iPhone o un iPad o un Mac, andate su iTunes, lì dalla piattaforma da cui scaricate canzoni e film, visitiate ICGED e troverete una serie di diverse centinaia di relazioni scientifiche a seminari seminari e lezioni, ma troverete anche l'intera collezione di Science and the City dell'anno scorso, che poi viene prodotta anche sotto formato di DVD e spedita a tutti gli istituti culturali italiani all'estero e a tutte le ambasciate e a moltissime scuole superiori in tutto il territorio nazionale, proprio per rappresentare una serie di eventi che contribuiscono un po' alla formazione culturale del nostro paese. Science and the City 2 e avrà luogo in queste giornate, praticamente una giornata alla settimana nel mese di aprile, si concluderà poi la prima settimana di maggio e si parlerà di argomenti molto vicini a tutti noi: fa bene, fa male vaccinarsi e a cosa servono i vaccini, perché c'è stato questo problema con l'influenza, è vera la storia dell'autismo, e poi è possibile rigenerare cervello,